Today our topic is anti-cancer drugs. The anti-cancer drugs, also known as anti-neoplastic drugs, work on different parts of cell cycle. In order to understand how chemotherapy drugs or agents work, we first have to revise cell cycle. The cell cycle has 4 phases: G1 phase, that is growth phase, S phase, G2 phase, and the M phase. In G1 phase, the organelles duplication occurs, and in the S phase, DNA replication occurs, and in G2 phase, cell prepares itself for the next phase, that is M phase or mitosis phase. In the M phase, where the cell divides into 2 daughter cells, then the cell cycle repeats. We can further explore mitosis into 4 more divisions, that is prophase, metaphase, anaphase, and telophase. In prophase, this is the phase where centrosome duplicates and form microtubules. In metaphase, the chromosome's DNA will align in the middle of the cell, and the microtubules that were formed from centrosome attach to the centromeres, which are the center points of the chromosome. In anaphase, the chromosomes are separated and reach either end of the cell. In telophase, the cell membrane constricts, ready to separate, and a new nuclear membrane is formed. The cell cycle is a continuous process, and so you have checkpoints to make sure that there are no abnormalities in the cell before it progresses to each phase. These checkpoints include G1 checkpoint, G2 checkpoint, and the M phase checkpoint. One thing these checkpoints look at is whether there are any abnormalities or damage or mutations to the DNA. For example, DNA is a double helix structure composed of 4 nucleotides. In the S phase, DNA becomes replicated. During replication, the DNA strand separated by the enzyme called helicase, as shown in the figure. During the unwinding of DNA, tension is created distally. Due to these tensions, coils are formed. The cells have a normal biological mechanism to fix this coiling and also other supercoils that have been formed. In this mechanism, there is an enzyme called topoisomerase II. Here, this enzyme called topoisomerase II fixes these coils and supercoils, hence reducing the tension in the DNA strand. There are 4 types of nucleotides present in DNA. These nucleotides can be divided into 2 groups: pyrimidines and purines. Pyrimidines include thymines and cytosine. Purines include adenine and guanine. So what happens when a double-stranded DNA gets unwound by helicase into 2 separate strips? Here another enzyme called DNA polymerase comes and creates a new strand on both the strips. The new strand following the helicase is called leading strand and the other is called lagging strand. Chemotherapy drugs target different parts of cell cycle. Because of this, they are grouped into different cases. One is cytotoxic drugs and the other is the drugs affecting hormonal milieu. Cytotoxic drugs. These can be further classified into alkylating agents, antimetabolites, CAMPOTHECIN analogues, antibiotics, taxanes, VINCA alkaloids, and miscellaneous. Alkylating agents. Alkylating agents work by alkylating DNA. Alkylation means the DNA is unable to replicate. It also means that DNA repair may result in DNA strands breaking apart. They form highly reactive intermediate compounds that are able to transfer alkyl groups to DNA. How alkylating agents kill cancerous cells? The answer is alkylating agents kill the cancerous cells by breaking the DNA strands. Alkylating agents work by reacting with the proteins inside the DNA of cancerous cells and make sure that they alter the very setting of cancerous cells. That is, these alkylating agents add an alkyl group to the proteins, thus preventing the proteins from linking up, causing breakage of DNA strands and eventually leads to death of the cell. Alkylating agents can be grouped into 5 categories: nitrogen mustard derivative, ethylenamine derivative, alkyl sulfonate derivative, and triazine derivative, nitrosourea derivative. Here you can remember these drugs classification as NEAT. N repeats 2 times. EAT, NEAT. These are the first letter of the words. Coming to nitrogen mustard derivatives. The drugs which come under this group are chlorambucil, cyclophosphamide, meclorethamine, and ifosfamide. Next, ethylenamine derivative, thiotepam. Alkyl sulfonative derivative, busulfan. Triazine derivative, dicarbazine. Nitrosourea derivative, carmestine and lomustine. Antimetabolites. Antimetabolite drugs were among the first effective chemotherapeutic agents discovered. These are classified as folic acid, pyrimidine, or purine analogues. These compounds have similar chemical structure to molecules that the body uses in nucleic acid synthesis, that is the DNA synthesis and RNA synthesis. Generally, antimetabolites induce cell death during the S phase of cell growth when incorporated into RNA and DNA and inhibit the enzymes needed for nucleic acid production. These agents are used for a variety of cancer therapies including leukemia, breast cancer, pancreatic cancer, ovarian cancer, and gastrointestinal cancers. Antimetabolites are divided into 3 subclasses, as we have seen before: folate antagonist, purine antagonist, pyrimidine antagonist. Methotrexate drug comes under folate antagonist. 6-mercaptopurine, 6-MP, fludarabine, 6-thioguanine, 6-TG, azathioprine come under purine antagonist. And then 5-fluorouracil, capecitabine, cytarabine, gemcitabine. Here you can see BINA, bine, the end of the drugs names. These are pyrimidine antagonists. Coming to folate antagonists. Actually, folic acid is a necessary compound for the production of nucleotides in the body. Intracellular folate is converted by the enzyme dihydrofolate reductase DHFR to dihydrofolate. This compound is then reduced to tetrahydrofolate. Tetrahydrofolate acts as carbon carrier that donates methyl groups to end target molecules through the enzymatic action of thymidine synthesis. Dihydrofolate reductase is continuously used in this process and is the site where the folate antagonists function. The drugs impede enzyme action and hence interfere with nucleotide formation. Methotrexate binds to dihydrofolate reversibly and inactivates it. This prevents methylation and decreases available supplies of purine and thymidine bases for new DNA and RNA synthesis. Methotrexate is active in the S phase of cell growth. It is used in many carcinomas: breast cancer, head and neck cancer, colorectal cancer, non-Hodgkin's lymphoma, osteosarcoma, bladder and choriocarcinoma. It is also used in acute lymphocytic leukemia and some types of meningeal carcinomas. Methotrexate is the most common folate antagonist used today, even though Other drugs have been developed. Purine antagonists. Purine analogues are antimetabolites that mimic the structure of metabolic purines. These are highly effective anti-neoplastic drugs. 6-mercaptopurine is the thiol analog of hypoxanthine. 6-mercaptopurine and 6-thioguanine were the first purine analogues to prove beneficial for treating neoplastic diseases. Azathioprine, an immunosuppressant, exerts its cytotoxic effects after the conversion to 6-mercaptopurine. Purine antagonists used for the treatment of malignant diseases, that is mercaptopurine and thioguanine. And for immune suppression, azathioprine. The purines, adenine and guanine, are chemicals used to build the nucleotides of DNA and RNA. The purine antagonists function by inhibiting DNA synthesis in two different ways. One is they can inhibit the production of purine containing nucleotides, adenine and guanine. If a cell does not have sufficient amount of purines, DNA synthesis is halted and it doesn't occur, and then the cell cannot divide. The next way is they may be incorporated into DNA molecule during DNA synthesis. The presence of these inhibitors is thought to interfere with further cell division. Purine antagonists currently used to treat cancer patients. These are 6-mercaptopurine and 6-thioguanine. These drugs are similar to each other and work in the same way. Pyrimidine antagonists. These act to block the synthesis of pyrimidine containing nucleotides cytosine and thymine in DNA, cytosine and uracil in RNA. These drugs are used to block the construction of these nucleotides. They have the structure similar to a natural compound. By acting as decoys, these drugs can prevent the production of finished nucleotides. They may exert their effects at different steps in that pathway and may directly inhibit crucial enzymes. The pyrimidine antagonist may also be incorporated in a growing DNA chain and lead to termination of the process. Camptothecin analogues. Topoisomerase is a major target for anti-cancer drug discovery and design. As a result, topo inhibitors constitute an important class of the current anti-cancer drugs. To date, all of the topo inhibitors that have been clinically evaluated are analogues of camptothecin, which is an extract of Chinese tree Campothica acuminate. This camptothecin has shown significant anti-tumor activity to lung cancer, ovarian cancer, breast cancer, pancreatic cancer, and stomach cancers. The drugs which come under this group are irinotecan and topotecan. Here tecan is the word that comes at the end of the drugs. Antibiotics. These chemotherapy medicines are called anti-tumor antibiotics. These antibiotics are not like the antibiotics that are used to treat other infections. Instead, these anti-tumor antibiotics interfere with the DNA inside the cells, which gets hampered in the process. This slows or stops cancer cells from growing and keeps them from multiplying. The drugs which are used are dactinomycin, doxorubicin, daunorubicin, bleomycin, mitomycin, mitoxantrone. Taxanes. The possible mechanism of action of taxanes is disruption of microtubule function. These microtubules are the organelles which are very essential for cell division, and the taxanes stabilize GDP bound tubulin in the microtubule, thereby inhibiting the process of cell division, and depolymerization is prevented. The taxanes are mitotic inhibitors, meaning that they inhibit tumors primarily by preventing cells from entering mitosis, which is a process of cell division. In addition, Taxanes appear to stimulate apoptosis. Paclitaxel and docetaxel are the drugs which come under this, and they act on ovarian cancer, breast cancer, lung cancer, Kaposi sarcoma, cervical and pancreatic cancer. Docetaxel acts on breast cancer, head and neck cancer, stomach cancer, prostate cancer, and non-small lung cancer. VINCA alkaloids. VINCA alkaloids are a set of antimitotic and antimicrotubule alkaloid agents originally derived from the periwinkle plant Catharanthus roseus, Vinca rosea, and other Vinca plants. They block beta-tubulin polymerization in a dividing cell. During cell division, Vinca alkaloid molecules bind to the building blocks of a protein called tubulin, inhibiting its formation. The drugs work during the mitotic phase of cell reproduction. Vincristine, vinblastine, and vinorelbine are the drugs. Here, at the beginning of the drugs, as these are Vinca alkaloids, VIN1 repeats. Miscellaneous drugs. These are procarbazine, etoposide, which is topoisomerase 2 inhibitor, hydroxyurea, and there is an important enzyme called L-asparginase, imatinib, nilotinib, sorafenib, bortezomib. Up to now, we have studied about the cytotoxic drugs. Next category is the drugs affecting the hormonal milieu. These are of two types, hormone responsive drugs and hormone dependent drugs. In hormone responsive category, the tumor regresses by hormone therapy. In hormone dependent category, the tumor regresses by the removal of hormone therapy. The drugs which come under this group are glucocorticoids, estrogens, progestins, selective estrogen receptor modulators, that are SERMs, tamoxifen, tormefen, selective estrogen receptor down regulators, SERDs, fulvestrant, then aromatase inhibitors, letrozole, anastrozole, exemestane, anti-androgens, flutamide, bicalutamide, 5-alpha reductase inhibitors, finasteride and dutasteride, then GnRH analogues, neferellin, triptorelin. Thank you.